So in this second part of the lecture, we'll talk about the cellular defenses in the immune system. So the innate and adaptive immune system include a variety of different immune cells that all have specialized functions. And these cells are produced through a process called hematopoiesis. Right? So think our bodies are composed of thousands of different types of cells. So those include our immune cells, but also cells that make up your skin and your liver and your heart. And all these cells have unique functions. Right? So how do our bodies develop from a single cell with all the same genes to the thousands of different types of cells that make up our body? So in your body, the way that all these cells come to be, that are in your body, you have stem cells. Stem cells are cells that can change and develop into many different types of cells, depending on what chemical signals and other triggers those cells receive. Right? During development of a person, so while you're growing up, those cells change and become differentiated into cells that make up all of the tissue in your body. Hematopoiesis is a process of development that occurs all throughout your life. Right? So you have stem cells around your body that can differentiate into all of the different cells that make up the cells found in your blood. So hematopoietic stem cells can differentiate into a variety of different cells that are found in the bloodstream, including erythrocytes. Those are red blood cells. They can differentiate into leukocytes. Those are what we call the white blood cells, or the immune cells. And they can also differentiate into thrombocytes. And so those are your platelets, or the cells that are responsible for helping your blood clot. So we're going to talk about the leukocytes specifically, those white blood cells that make up your immune system. Right? So the granulocytes are types of cells in your innate immune system that are produced during this process of differentiation in hematopoiesis. They include neutrophils, which are specialized immune cells that are responsible for killing extracellular bacteria by excreting chemicals, and also by phagocytizing or engulfing those bacteria. Another type of granulocyte are eosinophils. They protect against many eukaryotic pathogens, including protozoa and helminths, and they're also involved in a number of different autoimmune or allergic diseases. Basophils are cells that trigger inflammation and produce a chemical called histamine. Basophils are often involved in common allergic reactions that many of us experience. Mast cells have functions that are similar to basophils, but rather than circulating around the bloodstream, mast cells are permanently residing in the tissue around your body. So granulocytes are called granulocytes because they have these granule particles that are visible inside the cells if you view them under a microscope. And so these are these little particles that are shown in the cartoons and are a little bit visible on these blood smear images. The other characteristic of granulocytes that you see under microscopic examination is that they have these nuclei that are multi-lobed, and we can use these distinct features of these cells to look for immune pathologies in blood smear slides. Agrogranulocytes include a couple of different and important cells in the hematopoiesis process. They include immune sentinel cells, such as macrophages and dendritic cells. These are cells that monitor the body for pathogens. When they find a pathogen, they phagocytize those pathogens, or engulf them and chew them up. And then they chew up those pathogens in their chemical components and then present those chemicals as antigens to adaptive immune cells. So macrophages are immune sentinel cells. that present antigens mostly near the site of an infection. And dendritic cells are cells that act a lot like macrophages, but they also are responsible for migrating into the lymphatic system and presenting antigens to distant immune cells. So those macrophages and dendritic cells are also called antigen-presenting cells because their primary responsibility is presenting antigens to adaptive immune cells. Another type of agrogranulocyte are natural killer or NK cells. NK cells are responsible for destroying abnormal host cells. Right? So natural killer cells are found all over your body, and normally when they encounter another cell in your body, that cell sends a signal to the NK cells that say, I'm healthy, please don't kill me. Right? The NK cell recognizes this healthy cell signal with an inhibitory receptor, which is a protein that's present on the outside of the NK cell. Right? An activating receptor, on the other hand, is responsible for recognizing signals that are presented on the outside of cells that are behaving abnormally, such as cells that have been infected with a virus, or sometimes cells that are even cancerous or have other abnormalities. Right? When the NK cell doesn't get this inhibitory signal and does get the activating signal, then the NK cell releases a number of toxic chemicals from the cell to induce apoptosis to destroy the abnormal cell. So, how does the immune system respond when a pathogen enters the body? So, diapedesis is a process that your body undergoes when a pathogen breaches your epithelial tissue. Right? So if you get a cut in your skin and pathogens such as bacteria enter into your body through that cut, that macrophages that reside in tissue, and remember these are some of those immune sentinel or antigen presenting cells, they are hanging out in that underlying layer of skin tissue. They encounter that bacteria, or at least a few of those bacteria, and they phagocytize or engulf that bacteria. And when they do that, they also release a bunch of cytokines. So those are the molecules that signal to nearby and sometimes distant cells. Those cytokines then migrate into the blood vessels, and they attract other immune cells to the site of that infection. At this point, the blood capillaries, the blood vessels underneath the skin, they begin to dilate. Right? So they expand, and that allows these immune cells to fit through those normally tight junctions that make up the endothelial tissue. Right? Then the immune cells, they move from those capillaries, from the underlying blood vessels, into the surrounding tissue. Right? And once they're there, they work together to destroy that invading pathogen. And that process of those immune cells moving from the bloodstream into the surrounding tissue is called diapedesis. So, an important concept in how your body recognizes that there is a foreign pathogenic microbe in it is understanding molecular interactions. Right? So we've talked about a number of different molecular interactions already in this course. So molecular interactions including biological molecules that fit together because of their shape and the specific chemical properties that make up the atoms and molecules. We talked about DNA nucleotides that have the specific base pairing interaction, where hydrogen bond donors and acceptors form these complementary base pairs between A and T and C and G. We also talked about how enzymes catalyze chemical reactions by having a binding pocket that is a perfect chemical environment for the substrate to undergo a chemical reaction. And also by enzyme inhibitors that work by binding into that active site pocket. We also talked about virus spikes that are responsible for forming specific interactions with host cell receptors in order to induce the viral entry process. And in this lecture, we're going to talk about immune receptors that are responsible for identifying pathogens by binding to forming specific interactions with chemicals that are found on those pathogens. So microbial pathogens, bacteria, viruses, and protozoa and fungi, as we've talked about, they are made of different chemicals from humans, right? So for example, peptidoglycan is that chemical that we talked about that makes up bacteria cell walls. And so the peptidoglycan is a carbohydrate and amino acid polymer. We also talked about flagellin, which is a protein that's specifically found in bacteria flagella. And we've also talked about lipopolysaccharide, or LPS, which is a chemical that is exclusively found in the membranes of gram-negative bacteria. We've also talked about viruses that have double-stranded RNA or single-stranded RNA genomes. And those viral RNAs are different than RNAs that are found in our body. So these are unique chemicals. They're unique chemicals uniquely associated with pathogenic microbes. So we generally term these unique chemical patterns as pathogen-associated molecular patterns, or PAMPS. So how do our cells know that something is a pathogen? Well, it's because our immune cells have pattern recognition receptors, or PRRs, that are proteins that our immune cells and our bodies make to detect these unique chemical patterns that are unique to pathogens. So the PRRs detect the PAMPs by having a structure that perfectly fits or interacts with the PAMP chemical. Then the pattern recognition receptors, they trigger a reaction inside the cell in order to trigger an immune response. So often that involves the production of cytokines or signaling to nearby immune molecules. So one example of these pattern recognition receptors are toll-like receptors. So toll-like receptors are present in the membranes of many, many human cells. And so these are proteins that recognize many chemicals that are common to a variety of different pathogens. So some of these things that are recognized by toll-like receptors include bacteria flagellin, which is specifically recognized by toll-like receptor 5, lipopolysaccharide, which, as we said, is exclusively found in the outer membranes of gram-negative bacteria, and that is recognized by toll-like receptor 4. And then double-stranded RNA, which is not found in our body anywhere, but is found in viruses that have double-stranded RNA genomes. And so humans produce 13 different toll-like receptors, and so these 13 different toll-like receptors each recognize a unique chemical that is only found in a variety of different pathogens. Right? So these toll-like receptors are monitoring for these pathogen-associated patterns, and then they bind to those chemicals and then trigger an immune response. So when our immune cells recognize a pathogen, one of the things that many of our immune cells do is respond to that presence of that pathogen by undergoing phagocytosis. So phagocytosis is a mechanism where particles on the outside of cells are engulfed into a membrane compartment and then degraded inside of the cell. Right? So once that macrophage or other phagocytic cell engulfs, for example, a bacterium, then inside the cell that phagocyte is fused with another vesicle that includes enzymes such as lysozyme that are going to be able to degrade or break down that foreign pathogen. When these chemicals are broken down inside the cell, then that generates a set of broken down waste chemicals that are all unique chemicals that are only found in that pathogen. And these become antigens that are then going to trigger an adaptive immune response that we'll talk about briefly. So some pathogens are able to exploit this phagocytic process in order to get into the cell. And so instead of being degraded inside the cell, they sneak out of that phagosome. And you can read an example of this in the part in OpenStax chapter 17.4, when phagocytosis fails. Another way that our immune cells respond to pathogens is by producing inflammation. Right? So inflammation is a physiological response that is triggered by pathogen recognition. And so when we experience acute inflammation, when we have pattern recognition receptor stimulation, for example, acute inflammation can be triggered by those toad-like receptors. And that stimulation causes nearby inflammatory cells, such as mast cells, to produce histamine. So mast cells produce histamine, and this causes vasodilation, which is the widening of the blood vessels that increases blood flow. So many of your important immune cells are present in your bloodstream. They're those white blood cells. And so the increased blood flow then brings those immune cells to the site of the infection. The other thing that vasodilation does is it increases the permeability, and that allows those immune cells to go through the process of diapedesis, where they move from the blood vessels into the surrounding tissue. Right? So this acute inflammation floods the nearby tissue with immune cells and fluid. And that's why in this acute inflammation phase, you get swelling at the site of a cut or a bruise or other trigger of this acute inflammation process. Right? So acute inflammation is often painful because you have an increased amount of blood flow. Right? But it's usually, and I say usually, important for immune response. The reason I say usually is that we can, in many cases, experience chronic inflammation, where instead of having that inflammation last for only a short period of time and going away once the infection has been cleared, chronic inflammation is when the immune system fails to clear the infection, or it becomes overstimulated. And so there are cells that are sending out too many cytokines. Right? So chronic inflammation is constantly causing this vasodilation, so you experience painful swelling all the time. Those immune cells that are drawn to the site of inflammation, right, they produce a lot of chemicals in order to fight off the infection, but that's also damaging for our tissue. And so chronic inflammation can lead to long-term tissue damage. Right? So we talk about having a strong immune system and a good immune response, but really we don't want an immune response that is too strong, as we're going to talk about. We want an immune response that's just right. So we want inflammation to be triggered at the appropriate time, and then we want it to go away. And when our immune system is overactive, this can cause problems. Another physiological process that occurs during inflammation is fever. And so fever is controlled by the hypothalamus, which is a region in your brain that responds to the hormone prostaglandin. Right? And that signals the hypothalamus to raise your body temperature. So pyrogens are chemical signals produced by immune cells that stimulate this fever response. So as a clinical sign, a fever is defined as a body temperature that is above the normal range of 37 to 38 degrees Celsius. So fever rarely goes above 42 degrees Celsius, and temperatures that high can be dangerous. Right? Interestingly, we don't know exactly what fever does to help fight off infections. But our immune system does seem to work better when we do have a fever. On the other hand, that fever response also is causing you, a lot like the inflammation, to feel pretty sick. So having a fever usually makes you feel pretty awful. And taking fever-reducing medications, such as Tylenol or ibuprofen, can help you feel better when you have a short-term infection. Right? And so in most cases, it's okay to reduce the fever and reduce these inflammatory processes by taking things like ibuprofen.